Assalamu alaikum. I'm Dr. Cakaشه, Assistant Professor of Microbiology, and today we're going to start the first lecture of Microbiology in Infectious Disease 1 module. Today our topic title is Microbial World. By the end of this lecture, you will be able to categorize different microorganisms into prokaryotes and eukaryotes. You will be able to compare between prokaryotes and eukaryotes, classify medically important bacteria, discuss the normal microbial flora, and explain the relation between the two groups of normal flora. You should be able to list the beneficial role of resident flora and also list the body sites that contain normal flora. What's microbiology? Microbiology is the science of studying small, which is micro, living organisms. So it is the study of living things so small that they cannot be seen with the naked eye. Before the discovery of microscopes, this was not possible. So the discovery of the microscope made the science of created the science of microbiology because it allowed the visualization of structures too small to be seen by the naked eye. Microorganisms are a heterogeneous group of several distinct living structures of microscopic size. So they do not contain only a single type of organisms, but they are different different organisms that are very small to be seen, so they are gathered under the title of microorganisms. They include unicellular organisms such as bacteria, fungi, protozoa, and algae. Based on the on the difference in their cellular organization and biochemistry, these microorganisms have been classified into prokaryotes and eukaryotes. As you can see, this is a diagrammatic illustration of prokaryotes and eukaryotes. As you can see here, that prokaryotes are much simpler type of cell structure-wise, while eukaryotes are more complex, and we're going to talk more in details about the differences. Prokaryotes, bacteria, and blue-green algae are prokaryotes. They are bacteria as a unicellular free-living organisms. They have both DNA and RNA. They are capable of performing all essential processes of life, which include growth, reproduction, and metabolism. So although they are very simple structures, but they are capable of existing depending on themselves, and they can reproduce and grow, and they have different metabolic pathways which they need for their survival. While eukaryotes include fungi, algae other than the blue-green algae, which belongs to the prokaryotes, protozoa, molds are all eukaryotes. As we see, the eukaryotic cell is a much more complex type of cell. What is the difference between the two cells that we have seen? We'll enumerate the difference and we'll look back at the diagrams. As we see here, that prokaryotic cells don't have a nucleus, while eukaryotic cells do have a nucleus. DNA in eukaryotic cells are associated with histones. Histones are not present in prokaryotic cells. Organelles in the eukaryotic cells are surrounded by membranes, while in prokaryotic cells there are no membranes surrounding cellular organelles. In cell membrane composition in eukaryotic cells, they contain sterols, which is a type of lipids, while in the prokaryotic cells there are no sterols except in mycoplasma. The cell wall composition in prokaryotic cell is has a very specific type of molecule, which is a peptidoglycan. So peptidoglycan is present or specific for prokaryotes, while it's absent in eukaryotes. And the exception in the prokaryotes is also mycoplasma. So mycoplasma has sterols and doesn't have peptidoglycan, and this is the only exception in prokaryotic cells. Size of ribosomes is different from prokaryotes than eukaryotes. In prokaryotes it is 70S, while in eukaryotes it is 80S. Prokaryotes are always unicellular, while eukaryotes are multicellular. Okay? The size of cell is much smaller in prokaryotes than eukaryotes. It is from 1 to 5 micron, while in eukaryotes it is from 10 up to 100 microns. Prokaryotes do not have any kinds of mitotic division. They divide or reproduce by binary fission, while eukaryotic cells, they have mitotic division occurring in them. So if we look back at these different types of cells, we can see here that we have a specific well-defined nucleus, while we have here the arranged the DNA doesn't have, it's not membrane bound in the form of a nucleus. We have different organelles which are membrane bound. This doesn't exist in the prokaryotic cells. We have, as we said, we have cell wall and we have cell membrane. Cell membrane doesn't have sterols. Cell wall is specific, contains peptidoglycan. Here there is no peptidoglycan, and cell membranes contain sterols. Size difference, as we said, there is a major size difference. This can reach up to 100 microns, and this is the maximum is from 5 to 10 microns. So this is, as we said, is a much more complex structure. Okay, we go back. So now we're going to talk more specifically about bacteria. They form a large group of unicellular prokaryotic microorganisms. They vary in size from 0.1 micron up to 5, maximum 10 microns long. They have simple structure and they contain both DNA and RNA. Most medically important bacteria will grow on artificial culture media. What does this mean? This means that I can isolate this bacteria, there are a lot of bacteria existing and living around us in the soil, in water, as normal flora of different living beings, but and most of these bacteria, we only study the ones that are of clinical importance to humans, okay? The ones that cause disease or can impact the human health. Most of these bacteria, which are of clinical importance, most of them can be isolated. I can grow them in the lab on culture media. So most medically important bacteria will grow on artificial culture media in the lab, and this bacteria will reproduce by binary fission. What does this mean? This means that if I have a bacteria and I put it on a culture media, this is a petri dish which contains nutrient material, okay? The bacteria will start to multiply. It starts by one cell, and this cell will replicate its DNA. Okay, so it will have 2 copies of the DNA, and then, as we see, the cell will elongate, will divide in the middle, and produce 2 daughter cells. And this is how the bacteria replicates or reproduces divides by binary fission. So one parent cell will produce 2 identical daughter cells. This happens when the bacteria, I grow the bacteria in my petri dish, which contains nutrient media. The bacteria will start dividing by binary fission, and a single bacteria will reproduce by this way, or will divide by this way in a few minutes, and in a few hours or overnight, if I incubate it in this plate in 37 degrees, so I provided my bacteria with proper temperature, I give it nutrients, okay, by providing media which contain nutrients for the bacteria, and give it time to grow and reproduce. So the bacteria will divide over hours, okay, and will produce millions of bacteria. By the end of my incubation, I will be able to see these small colonies. So each colony of this is composed of millions of bacteria. A single bacteria was able to divide over hours, okay? Each division takes only minutes, so it was able to divide over hours, producing millions of bacteria that it formed a colony. This colony is now visible to my naked eye. I can see the colonies with my naked eyes, okay? So what I can see here, each dot of this is a colony of bacteria representing millions of bacteria. So bacteria can be classified according to their morphology, according to their shape. How do they look like? I can divide them into groups according to their staining reactions. Okay, do they take what type of stain I can stain them with? I can classify them according to other features, including the ability to grow in the presence of oxygen or in the absence of oxygen. Are they aerobic or anaerobic? I can also classify them, for example, according to spore formation. Are they able to form spores or not? And all of these, you will take them in details in upcoming lectures. If I want to classify bacteria according to their shapes, you'll find that bacteria can be spherical, like a circle, and this is what we call cocci or a coccus. Okay, so bacteria mainly may be spherical, and this is what we call cocci. They might be a little bit elongated. This is oval shaped, and this is what we call coccobacilli. Okay, so we have cocci, we have coccobacilli. Okay, when it's elongated. If it is rod shaped, okay, we call it a bacilli. That's why when it is somewhere in between the cocci and the bacilli, we call it a coccobacilli. Okay, it is curved, we call it, it's comma shaped. This is vibrio. It can be spiral in shape, and we have two examples like the spirillum and spirochetes. So these are different morphologies for the bacteria. The most common that we will see are the cocci and the bacilli. So these are the most common that we will see under the microscope, or our bacteria are divided into mainly cocci and bacilli, but we have others that exist in different shapes in between, like slightly elongated cocci, we call it coccobacilli. It's somewhere in between, or curved or comma shaped, which are the vibrios, or they can be spiral in shape like spirochetes and spirillum. So these are classifying the bacteria according to morphology, not only according according to the shape, also according to the arrangement. So a cocci can present, a coccus, which is bacteria which has a spherical shape, can present as single cocci, okay, or they can be arranged as pairs. Okay, they are arranged as pairs, as we can see here. We call them diplococci because they are two. Okay, as we see here, we have arrangement in pairs. We have examples here. It is Streptococcus pneumoniae, and this is another bacteria called Neisseria. These two types of bacteria are arranged in pairs. Okay, they can be arranged in tetrads, fours. Each four they come together and they are arranged in tetrads. Okay, they can be arranged in chains, one after the other. They form a chain, and the example is Streptococci. They can be arranged in clusters like grapes. We call them grape-like clusters, and this group is Staphylococci. So we don't only have a specific shape for the bacteria might also have a specific arrangement. Okay, this arrangement for the cocci, for example, they can be pairs, or they can be in groups, whether they are in fours, or we have here an example in packets, okay, or in grape-like clusters, or in chains. Don't worry about the names or examples of the bacteria, because you will take them more in details later on, and they will become much more easier as we go on with different lectures. What about the bacilli? The bacilli can also have specific arrangement. Some of them might be arranged, as you can see, in chain. We can have the bacilli. We can have, of course, as we said, a little bit somewhere in between, which are oval shaped. We call them coccobacilli. Some bacilli might be arranged in palisades. What are palisades? Palisades are when the bacilli are arranged at angles to each other. They form angles together, or they are arranged parallel to each other, and this is what we call a palisade arrangement. Our example is Corynebacterium. Okay, some bacteria, as we said, bacilli might have spores. They are able to form spores, and when we see this, then we know that they are spore-forming bacteria. Some of them might be filamentous. Okay, they are very tall and elongated, appear as if they are branching. Okay, and these we can call them filamentous bacteria. So in the bacilli, as an arrangement for the bacilli, they can have a palisade arrangement. They can have a short chain-like arrangement, or they can be elongated and filamentous. Something specific also for the bacilli is that some groups are able to form spores inside the bacilli. So as we said, these are the two main shapes, cocci and bacilli, two main morphologies, and these are specific arrangements for both the cocci and the bacilli. We have other shapes which we talked about. We have the curved or the comma shaped like the vibrios, and we have the spiral shapes like the spirillum and the spirochetes. Okay, so we also, as we said, we can classify our bacteria according to their shapes and arrangement. This will help us to identify the bacteria. So if I look under the microscope, I find that this is a cocci arranged in chains, then this is most probably streptococci. If it's arranged in grape-like clusters, then this is most probably staphylococci. This is, of course, beside the Gram stain, which we're going to talk about. So we said we can classify the bacteria according to shapes and arrangement. Okay, so we can classify the bacteria according to their morphology, which includes the shape and arrangement. We can classify the bacteria according to the staining reaction. So what does this mean? Means that we have different stains, or we have different stain, but there is an important specific stain for bacteria. Okay, we have an important specific stain for bacteria called a Gram stain. As we see here, a Gram stain. What is a Gram stain? The Gram stain was named after the person who invented it. This is Gram, and he used a specific type of staining procedure that contains two different stains. Okay, a violet stain and a pink stain. The bacteria that took, and you will take this in details in the practical class later on and in further lectures. The bacteria that took the violet color, we called it gram-positive bacteria. And the bacteria that took the pink color, we called it gram-negative bacteria. So the bacteria are now classified according to the Gram stain into gram-positive and gram-negative. Gram-positive will appear under the microscope as violet color, and gram-negative will appear under the microscope as pink in color. And as we said, we can classify the bacteria also according to its shape. So now I have gram-positive cocci, okay, so cocci that appear violet in color when stained with the Gram stain. I have gram-positive bacilli. These are bacilli that will appear violet in color under the microscope when stained with the Gram stain. I can have gram-negative cocci and bacilli. Gram-negative cocci, they will appear pink in color under the microscope, and gram-negative bacilli will also appear pink in color. So medically important bacteria, according to the staining characters, can be by the Gram stain, can be classified into gram-positive bacteria and gram-negative bacteria. The gram-positive bacteria take the violet color in the Gram stain, and the gram-negative will take the pink color in the Gram stain, and you will take the details of the Gram stain in your practical class and further lectures. Majority of the bacteria are stained by Gram stain, but we have others that do not take the Gram stain, like the Mycobacterium and Spirochetes, and we can see them using other techniques, other stains and other types of microscopes. And we have atypical Mycobacteria, Chlamydia and Mycoplasma and Rickettsia, and these are also seen by other staining or other microscopic procedure. But most of the bacteria stained by Gram stain are divided into Gram-positive and Gram-negative. I will read with you the names so that you find them easier to study, but as I said, if you will get to know them further on as we take on the lectures. Don't worry if you don't learn them by heart from the start. The gram-positive bacteria cocci include staphylococci, streptococci, and enterococci. Okay, the bacilli, the gram-positive bacilli include Corynebacterium, Bacillus, and Clostridium. The gram-negative cocci and coccobacilli include Neisseria haemophilus, Bordetella, and Brucella. The gram-negative bacilli include Enterobacteriaceae, which are a large group of gram-negative bacilli, Pseudomonas, Vibrio, Helicobacter, and Campylobacter. So now we have divided the bacteria according to the morphology, classified the bacteria according to morphology, and according to the staining character. Okay, now we're going to talk about the normal microbial flora. So we have bacteria existing everywhere in the environment, and also they exist living with us on our body surfaces. Okay, so microbes that engage in mutual or commensal association with humans are called the normal flora. So bacteria that coexist with us, living on our surfaces, whether skin or mucous membrane surfaces, these are called our normal flora. They are normal, we coexist, and we know them as commensals. Okay, they live with us and they don't cause problems. These organisms live in complete harmony with the host without causing any damage to it. We have to remember that the bacteria are enormous in number, the normal flora, that they exceed the number of our own living cells. The ratio of bacteria to human cell is 10 to 1 in a single person. In a healthy person with an intact immunological and anatomical defenses, so it doesn't have breaks in the skin, doesn't have ulcers on the mucous membranes. So in a healthy person with with a good immune response and proper anatomical defenses, these commensals or normal flora, such organisms rarely cause disease. Okay, except only under certain conditions, when a suitable opportunity arises. So if there is a suitable opportunity that comes to this bacteria that usually does not cause disease arises, it will cause an infection. In this case, there is an opportunity for this bacteria, so this bacteria becomes an opportunistic pathogen. Usually it's not a pathogen. Until the opportunity arises, it will become an opportunistic pathogen. For example, we have a bacteria known as one of the Enterobacteriaceae, a bacteria known as E. coli. This E. coli is normal flora of the gastrointestinal tract. Means that it is in the gastrointestinal tract there's a lot of bacteria. E. coli is one of them. They exist with us inside the gastrointestinal tract without causing any problems. If this E. coli reaches the urinary tract, this is not the normal site for it, it will cause a urinary tract infection. Okay, I have staph on my skin, for example, and it exists with me without causing problems. If I have an injury in my skin and this staph, and I do not properly disinfect my wound and cover it and give it and do proper disinfection and change my dressing and so on, then this staph will get an opportunity when it finds this break to multiply and inside this wound, okay, and will become an opportunistic pathogen and will cause a wound infection. Okay, so when an opportunity arises, the bacteria might cause an infection. In this case, it's an opportunistic pathogen. Do we have different types of normal flora? Yes, we have different types of normal flora. Normal flora can be resident or transient. Okay, so we have two groups, resident normal flora and we have transient normal flora. From their names, resident flora, these are the ones with permanent residences. They are permanent. They are usually, these are my flora, they coexist with me, they belong to me. Okay, transient flora, these are the flora which I acquire, and they are they are only visitors. Okay, so this is the difference between resident and transient flora. So the resident flora are those camping on living on my skin or mucous membrane. Transient bacteria are those passing by. Where do we acquire the transient bacteria from? So the resident are organisms which are regularly present in a given area at a given age. When disturbed, it soon re-establishes itself. I cannot get rid of my resident bacterial flora, okay? So when the count decreases, it will increase again. Okay, so when I wash my hands, for example, properly, or when I disinfect my hands with alcohol, the count of the resident flora, it will not disappear, will decrease, but they will multiply and they will re-establish themselves again on in this area. This is an example is Escherichia coli, which is E. coli found in the normal flora of the intestine, and Staphylococcus epidermidis found on the skin. These are examples of my normal resident flora. And we have alpha hemolytic streptococci found in the oropharynx. This is an example also of normal flora. What about the transient? The transient, this consists of both pathogens and non-pathogens that inhabit skin or mucous membrane for a limited period. They are not part of the normal flora, but I acquire them from the environment. If the resident flora is intact, there is a very little significance of the transient flora. Again, so I have my resident flora on my skin, for example. I touch this surface, I touch any surface. This surface contains bacteria that might not belong to, not the same as my resident flora. This bacteria will come on my skin. These are transient. These bacteria can be pathogenic or non-pathogenic. That's why hand washing is important, okay? So it can be pathogenic or non-pathogenic, okay? This is not their normal place, okay? If my normal flora, my resident flora is intact, okay, so I have the normal flora on my skin, it will give little chance for this transient flora to colonize my skin, to increase in number on my skin, for example. So if the resident flora is intact, there is very little significance of the transient for the transient flora. However, if the normal resident flora is disturbed, okay, the transient flora may have a chance to colonize, increase in number, proliferate, and become the main bacteria in this area. And if they are pathogenic, then they might be able to cause disease. So actually, my normal flora, my normal resident flora is a defense mechanism because it occupies the space on my skin or on my mucous membrane and doesn't give a large chance for any transient flora, pathogenic transient flora, to increase in number or produce disease. So what is the beneficial role? This brings us to the beneficial role of the resident flora. They suppress the colonization or invasion of the body by pathogens, by bacterial interference. What do we mean by bacterial interference? They compete with this visitor or transient bacteria for nutrients, for space. They do not give it a chance to increase in number because they already are established in this area. Okay, so they do not suppress the colonization of the transient flora, especially if they are pathogens. They do not give them a chance to increase in number. We have, as we said, a normal flora in the intestinal tract. Members of the intestinal flora are very important. They help in the synthesis of some vitamins, especially vitamin K, some vitamin Bs, and others, some other bacteria aid in the digestion of food. So intestinal bacterial flora is important because it helps in the synthesis of vitamins B and K, and they help in the digestion of food. They can immunize the host against pathogen when related or cross-reactant antigens are shared, thus raising the overall immune status of the host. So because our body is exposed to different antigenic or different bacteria which are part of the normal flora, this might give them immunity to other bacteria invading the body if they have similar composition. So this is the beneficial role of resident flora, protects the body from colonization by any transient pathogenic flora. They help in the GIT, the normal flora of the GIT helps in the synthesis of vitamins and in the digestion of food. And they can, being exposed to the normal flora will give a chance to our immune system to respond to other pathogens having similar composition to these bacteria. What are the sites of normal flora? Where can I find the normal flora in our body? Areas that are exposed to the environment are usually colonized with the normal flora. So we have skin and mucous membrane always carry a variety of organisms because they are in contact with the environment. Where do we have skin and mucous membranes? When we start, we have conjunctiva is exposed to the environment, so it will contain normal flora. The nasal cavity exposed to the environment contains normal flora. The airways, upper respiratory, like the oropharynx, also contains normal flora. The GIT is open from the top till the end. The GIT is all colonized with gastrointestinal tract, contains normal flora. Okay? So the intestine, the vagina contains normal flora, and of course the skin, because it's exposed to the outside, contains normal flora. Okay, so we have all surfaces and mucous membranes that are exposed to the outside will contain normal flora. But all internal organs, except the gastrointestinal tract and body fluids, are normally sterile. So any internal organ is considered sterile. So the lower respiratory tract, okay, not the upper, the lower respiratory tract is sterile. Okay, all other body cavities, for example, the peritoneum, the peritoneal cavity, this is considered sterile. All internal organs are sterile. The urinary tract, okay, is sterile. Urine is sterile. Our blood is sterile. Nervous system, CSF, sterile. Does not normally contain bacteria. If any bacteria reaches a sterile site, even if it's part of the normal flora, it will cause, okay, and it has a chance to multiply in number. Immune system is not strong enough. If the immune system is not strong enough to get rid of it, or it reaches this place in a large number, it might cause a disease. Okay? As the examples we said, that E. coli is part of the normal flora of the gastrointestinal tract. However, if it reaches the urinary tract, it will cause a urinary tract infection. The total number, as I told you, the total number of flora of the body contains more than 100 distinct species of microorganisms, and these microbes on the human body outnumber the human living cells. We have a lot of flora, bacterial flora, that coexist with us. These are examples of sites and names of bacteria that exist in these sites as normal flora, okay? But only in these sites. For example, bacteroid species, these are anaerobic bacteria. They are normally found in colon, throat, and vagina. Candida albicans found in mouth, colon, and vagina. So Candida, it's okay to find Candida in vagina, colon, or the mouth, but I cannot, for example, isolate it from the blood. Okay, this is not normal. Okay, Clostridium species are found in the colon. Corynebacterium, nasopharynx and vagina. Enterococcus and also in the skin. So I find that skin, for example, here the vagina contains Corynebacterium, contains Candida and bacteroid species. These are all part of the normal flora of the vagina. Okay, Enterococcus fecalis is found in the colon, so the colon contains, as part of the normal flora, Enterococcus, Clostridium, and bacteroids. Is it important to know the normal flora of a certain area? Yes, because this will help me if I get a report with certain names of bacteria. I know that these are normal flora. They are not pathogenic in this area. If I find them in another area that is normally sterile, then I will understand that these are probably pathogens, okay? Or that they can cause disease outside this area. E. coli and other coliforms are found in the colon and the vagina. Haemophilus in the nasopharynx and conjunctiva. I will go through the names for you just to make them easier when you read them. Lactobacillus species, we have Neisseria species, Staphylococcus aureus, Staph epidermidis, and Viridans streptococci. As I said, do not worry about learning the names from this lecture by heart. As we go through, you will learn more about each bacteria, and the names and the bacteria will be more easier to learn. Thank you very much. Now we come to the end of our lecture. Thank you.